Hello and welcome to my YouTube channel, AnatomyMentor. Today we're going to be discussing this topic, which is the histology of the nerves and the ganglia. So in today's discussion, we are going to be explaining the histomorphological composition of the peripheral nerve, and we shall explain myelinated, unmyelinated fibers of the central and peripheral nervous systems. Also, I'm going to be talking about the ganglia, the sensory and autonomic ganglia, and also a little bit of the clinical application in the discussion of multiple sclerosis and GB syndrome. So these are our learning objectives for today. And starting with today's topic, that is the first thing I'm going to talk about is a peripheral nerve. Now, a peripheral nerve is, you can define it as a bundle of nerve fibers which are held together by connective tissue. And what is a nerve fiber? This is the axon with or without its covering of myelin. And the connective tissue which is supporting this peripheral nerve, it can be further subdivided into three parts. That is the endoneurium, the perineurium, and the epineurium. So here is a diagram, and this is a very simple basic diagram which is showing us the distribution of connective tissue that is supporting a peripheral nerve. Over here, let's just focus first of all at a single axon which is surrounded by myelin sheath, and then comes its supporting connective tissue which is the endoneurium. So endoneurium is the loose connective tissue which is supporting a single nerve fiber. And now several of these nerve fibers, they are grouped together as bundles, also known as the fascicle. So the fascicle is surrounded by connective tissue, which is a specialized connective tissue, and this is known as the perineurium. And then several of these fascicles, they are collectively going to form the peripheral nerve. And the peripheral nerve as a whole is surrounded by this here tough layer of dense connective tissue, which is the epineurium. And epineurium also extends between the fascicles. Now let's look at these connective tissue elements individually. First, the endoneurium, which is the thin layer of connective tissue surrounding the individual axon or nerve fiber. It lies immediately adjacent to the external lamina of the Schwann cells. And it consists of reticular fibers, fibroblasts, and capillaries. Now here are the histomicrographs, the hematoxylin and eosine stain sections of the peripheral nerve, which is going to show us the relation between these three types of supporting tissue. So here on the left histomicrograph, you can see that we have the epineurium, which is the thickest, irregular, dense connective tissue surrounding the peripheral nerve as a whole. And then you can see these are the bundles of nerve fibers, and these bundles are surrounded by this connective tissue layer, which is the perineurium. It has been labeled as P in this histomicrograph. And if we look at this individual fasciculus at a higher magnification, then we can see it is consisting of these nerve fibers, and each nerve fiber is surrounded by the delicate, thin connective tissue layer, which is the endoneurium. And now let's move on and talk a little about the perineurium. The perineurium is a specialized connective tissue. Why is it specialized? Because it consists of these flat fibrocytes with their edges tightly attached to one another by the tight junctions, and several layers of these cells make up the blood nerve barrier that helps to maintain the fiber's microenvironment. So this is a special function of the perineurium. Now, if we look at the ultrastructure, that is through the electron micrograph, this is the electron micrograph taken through the scanning electron micrography. Over here, we can see that these are the axons, and these axons, they are surrounded by the endoneurium. The whole bundle of axons is surrounded by this here, perineurium. And here, the section is also through the blood vessels, which are visible. So the blood-nerve barrier, remember, is formed by the specialized cells lying within the perineurium. And this regulates the kind of substances in the plasma which are going to be released into the endoneurium. Okay, now the third one, which is the thickest, the dense layer surrounding the nerve as a whole, the perineurium. This is the one which is the tough one, and it also sends septi in between the bundles of the axons. Another electron micrograph, this is now taken through the transmission electron micrography. This is showing us over here, you can see F is labeling the fibroblasts surrounded by C, which is the collagen in the epineurium, that is the outermost layer surrounding the nerve. And then we can see over here, we've got the perineurium. And then these are the individual axons. Each axon is surrounded by this dark colored layer, which is the myelin layer. And this here axon, you can also see adjacent to it is the Schwann cell, nucleus, and cytoplasm. And the endoneurium, which is surrounding individual axons, that is consisting of the reticulin fibers, right? And also you can see a few axons which are unmyelinated axons as well in this cross section. Now, in the peripheral nervous system, remember all the axons are enveloped by the highly specialized Schwann cells, which are also called neurolemocytes. And these Schwann cells or neurolemocytes, they provide a structural and functional and metabolic support to the nerve fibers. Now, in the peripheral nervous system, first we're going to look at those nerve fibers which are the non-myelinated ones, and then we'll talk in detail about the myelinated fibers. So first, let's talk about those fibers which are said to be non-myelinated. Now, although they don't have the myelin covering, but they are supported by Schwann cells. So this is an important thing to understand, that these nerve fibers, they are lying within these clefts, or you could say troughs, which are present within the cytoplasm of this large size Schwann cell. So they are said to be engulfed, or you could simply say they are just lying within the cytoplasm of the Schwann cell. The zone of apposition of the two adjacent membranes of each Schwann cell, that is known as the MES axon. This effectively seals the axon, this here axon, in an extracellular compartment which is bounded by the Schwann cell. So unmyelinated axons, you can see over here, these are closely associated with the Schwann cell, and they're lying within these troughs or clefts of the Schwann cell. So within one Schwann cell, you can see over here that these are several axons, and they are lying within the troughs of a single Schwann cell. And remember, this is true for the small diameter axons, because these are the ones which are non-myelinated. But the wrapping process of myelination, that occurs in the large diameter axons, and those fibers are then said to be the myelinated nerve fibers. So what happens over here is that in this case, the first difference we have to remember is that a single Schwann cell is going to wrap itself around a single axon. And you can see over here that this wrapping process of the membrane and cytoplasm of the Schwann cell produces a thick covering which is surrounding this axon, and this insulating layer is now known as the myelin, and this nerve fiber is now said to be the myelinated nerve fiber. And over here, you can see that these are the points which are known as the nodes of Ranvier. These are the points between two internode regions. And what is an internode? It's a single segment of myelin produced by each Schwann cell. And between it, you can see this is the node of Ranvier. Here you can see the multiple membranous layers of the Schwann cell that have formed the myelin sheath. And on the outer aspect will now be present the Schwann cell nucleus. Also, you can see the Schmidt-Lantermann clefts. We will be talking about them in the next slides. So if we look at the ultrastructural features, then that is through the electron microscope, we have to look at some significant points The first is the major dense line. This represents the fused protein-rich cytoplasmic surfaces of the Schwann cell membrane. Then there is the intraperiod line. This is a faintly stained layer that represents the opposed outer bilayers of the Schwann cell membrane. And then come the Schmidt-Lantermann clefts, also known as the myelin incisures or clefts. These are certain points where the myelin sheath appears to be, you could say, disrupted. You see, the Schwann cell cytoplasm is retained at these points. So these uncompacted zones or regions are known as the Schmidt-Lantermann clefts. And in the longitudinal section of an electron micrograph, they pass obliquely across the width of the myelin sheath. So first, we're going to look at the major dense line and the intraperiod line. Like I told you, this here is showing you in these diagrams the process of myelination. This is a single axon, and the Schwann cell is wrapping its membrane around this axon, and these multiple wrappings have then formed the thick myelin sheath. Now, if we look at these, we can see a darker line and a lighter line. And remember, these are appreciable in an electron micrograph. The darker line is known as the major dense line. And over here, the cytoplasmic surfaces are opposed, whereas the lighter or fainter one, the intraperiod line, is showing the apposition of the outer bilayered Schwann cell membrane. Also, we should look over here at the outer mesoxon and the inner mesoxon. Now we're going to look at the electron micrograph and locate the Schmidt-Lantermann clefts. So you can see over here is the region of the Schwann cell cytoplasm, that is the peripheral Schwann cell cytoplasm, which is S1. And here is S2, which is the region of the, this over here you can see is the periaxonal Schwann cell cytoplasm. If you follow the pointer, the laser pointer over here, this oblique cleft you can see, which are the spaces actually representing the retained Schwann cell cytoplasm. This is an oblique cleft which is passing through the myelin sheath. This is known as the Schmidt-Lantermann clefts or the myelin incisures. And this here as a whole, this was the myelin sheath or covering surrounding this here axon. Okay, so if we are going to look at a teased preparation on the left of the peripheral nerves, which have been stained by Sudan black, you can see the myelin which is surrounding each axon. This myelin takes on a dark black color. And this here arrow is pointing towards the node of Ranvier. And let's look over here at the histomicrograph B, which is showing us the hematoxylin and eosine stain section at a medium power of the peripheral nerve. This is a longitudinal section. In this case, you can appreciate also the node of Ranvier. And you can see the nuclei, which are visible over here, will be of two types. The flattened ones are the nuclei of the fibroblasts present between the nerve fibers, and slightly more ovalish nucleus will be that of the Schwann cell. Now here is a comparison of peripheral nerves through two planes of section. On the left, you see the transverse or the cross section, and on the right, you can see the longitudinal section. If we look at the cross section, and remember this is a special stain, which is the toluidine blue stain. If we look at the left, we can see multiple axons which have been sliced through. A is representing axon, and it is surrounded by this thick layer of the myelin. But there are also some axons over here. You can see this double layer, which is representing the section which has been taken through the Schmidt-Lantermann cleft. Okay. And on the right, the longitudinal section of these same axons surrounded by the myelin layer. And you can also see these over here, the Schmidt-Lantermann clefts in the longitudinal section. Now, this is an electron micrograph, and this is the ultrastructure of both myelinated and unmyelinated nerve fibers. So first, let's look at the myelinated one. Here is the axon, and it is surrounded by this here thick myelin sheath. So this is the myelinated axon, and alongside it, you can see this, the massive, the large Schwann cell cytoplasm, the Schwann cell nucleus. And you can also see a few unmyelinated. If you look over here, this is an unmyelinated axon, it is lying in a cleft or trough of the Schwann cell. But as you can see over here, the myelin sheath is not present. So that is the unmyelinated axon. This is a simple diagram, which is just to help you formulate a three-dimensional concept of the myelination. This is an axon, and here the myelin sheath has been opened or unwrapped to show us how myelin itself is going to be arranged around that myelinated fiber. And in between the gaps, you can see where myelin is not present. These are the Schmidt-Lantermann clefts. So now let's come to the central nervous system. We have discussed the peripheral nerves. Now we're going to talk about the central nervous system. Over here, myelination is similar to that in the peripheral nervous system, but this function is going to be performed not by Schwann cells, but by another cell population, which are the oligodendrocytes. And the difference in this case is that oligodendrocytes, one oligodendrocyte is going to form myelin around several axons. So here is a single oligodendrocyte, and it is, you can see, myelinating several axons over here. But the process of myelination and the layer of the myelin, the myelin sheath itself, that is similar as was in the case of the myelination by Schwann cells. So remember that the process of myelination increases the conduction velocity through the nerve fiber. So, of course, one factor is the diameter of the axon. The larger the diameter, the greater is the conduction velocity of an action potential through that nerve fiber. But myelination also increases the axon conduction velocity. That is, it has an insulation property, and it also increases the conduction velocity. Now, let's apply this knowledge of histology to the clinical conditions that is known as the peripheral neuropathy. You see, there are two main patterns of peripheral nerve disease. In one case, the demyelination occurs, that is, the myelin sheath is lost, and that is known as the demyelinating neuropathy. And in the other type, the myelin sheath is not lost, but the axon itself is damaged, and that is known as an axonal neuropathy. Well, if we're talking about the disorders of myelin, then these are the two conditions which you should be familiar with. One is the multiple sclerosis, and the second one is the Guillian Barre syndrome or GP syndrome. Both these conditions are immune-mediated, and they consist of a demyelination, that is, the myelin sheath, which is surrounding the nerve fibers, is lost. The difference is that in case of multiple sclerosis, this demyelination occurs within the central nervous system, whereas in GP syndrome, this demyelination occurs in the peripheral nervous system. So the symptoms depends upon the nerve fibers which have been involved, and that leads to neurological dysfunction. In case of GB syndrome, there is a progressive weakness of limbs and weakness of respiratory muscles. And gene mutation also, remember, has a role to play in this in certain diseases, those which are the inherited disorders of the nervous system. Now that we've discussed the nerves, the histology of the nerves, we are now going to be talking a little about the ganglia. Now, what is a ganglia? What is a ganglion? Singular is ganglion, plural is ganglia. These are the collections or discrete aggregations of neuronal cell bodies located outside the central nervous system. So remember that the collection of neurons within the central nervous system is known as nuclei, but collection of neurons outside the central nervous system are known as ganglia. The neurons of ganglia themselves are surrounded by supporting glial cells known as the satellite cells, and also delicate connective tissue. But a denser connective tissue capsule is also present on the outer aspect of the ganglion in most cases. And the ganglia serve as relay stations for transmission of nerve impulses. So here is the basic classification of the type of ganglia. You can see they can be either sensory or autonomic. And the sensory are further subdivided into cranial or spinal. And when we talk about autonomic ganglia, they will be either parasympathetic or sympathetic. We're looking at a histomicrograph, which is of the spinal ganglion, and on the left is the H&E hematoxylin eosine stain section at a medium power. On the right is the hematoxylin eosine stain section at a higher power. So first, if we look at the spinal ganglion at a lower power, we shall see externally it is surrounded by this thick, dense connective tissue layer, which is forming a prominent, well-defined capsule on the outer aspect. And this spinal ganglion, remember, is present in the dorsal root of the spinal nerves. And you can also see the fasciculi, or you can say the septi, which are then emerging from this outer connective tissue capsule towards the inner aspect. And here, let's look at a higher magnification of the neurons which are present in this ganglia. These are the pseudo-unipolar neurons. Well, pseudo-unipolar means that they have a single process which then divides into two. And you can see over here that these pseudo-unipolar neurons, they are present, and outside the pseudo-unipolar neurons are the satellite cells. These are the supporting glial cells. Their nuclei, you can see, are present on the external aspect of the pseudo-unipolar neuron. So remember, these spinal ganglia, they are present in the dorsal root of the spinal nerve. Now let's come to the autonomic ganglia. You should keep in mind the basic organization of the autonomic nervous system. This consists of the preganglionic neurons with the cell bodies in central nervous system. From the cell bodies extend the myelinated preganglionic nerve fibers. They synapse on the postganglionic neuron, which has its cell body in the ganglion. And from there, the postganglionic non-myelinated nerve fibers will then project to the target. So this here is the ganglion, which is the autonomic ganglion, and it has the postganglionic neuronal cell bodies. Now let's look at the histomicrograph of this autonomic ganglion, which can, in this case, be sympathetic ganglion. These are multipolar neurons, and you can see these are, when we say multipolar, we mean they have multiple branches and they have multiple processes. And the nucleus within them is characteristically off-center, also known as the eccentric nucleus. And you can also see this is also surrounded by the supporting, the nuclei are visible, of the glial satellite cells. And also you can see over here is the lipofuscin pigment, which is the wear and tear pigment. It's a brownish pigment which accumulates over time within these neurons. So here is now the parasympathetic ganglion, which is also, this is another type of autonomic ganglion. These ganglia are typically present within the walls of the viscera. So you can see on the outer aspect, their connective tissue capsule is not so obvious, since the connective tissue simply merges or unites with the connective tissue of the viscus. So there's a poorly defined capsule. Okay, so now that we have discussed the essentials of the histology of the nerves and the ganglia, let's look at a few MCQs to reinforce what we just studied. So which of the following is the characteristic of the connective tissue layer that surrounds individual fascicles in large peripheral nerves? So which of these is a characteristic? Now, that connective tissue layer that surrounds individual fascicles or bundles in large peripheral nerves, that is the perineurium. And perineurium has the characteristic is that it has specialized cells which have tight junctions between them, and they form a seal known as the blood nerve barrier. So this is a special function of the perineurium. So C is the correct answer in this MCQ. Whereas if we look at the other options, a delicate region of connective tissue in contact with Schwann cells, that would be the endoneurium, not the perineurium. And of course, this is not dura mater, and it is not the myelinated area, because myelin is present around individual axons. And the thickest sheath of connective tissue, well, that is epineurium. Then again, it is not perineurium. So the correct answer is C, which is the quality of perineurium, which was asked in this MCQ. Now, what term applies to collections of neuronal cell bodies in the central nervous system? I told you during the discussion that the collection of neurons within central nervous system are known as nuclei. If they had asked about the collection of neurons in the peripheral nervous system, then the answer would have been ganglia. But since they're asking about the collection of neuronal cell bodies in central nervous system, the answer is D, which is nuclei. Now, this is a clinical scenario. A 36-year-old woman, she completes a 4-week medical mission to Brazil. 18 months after her return, she complains of loss of sensation in her hands and feet. So she has distal anesthesia. Neurologic examination reveals loss of temperature, light touch, pain, and deep pressure on her hands and feet. So these are the sensations that have been lost. A hypopigmented macula is present on the dorsum of the hand. So she also has a lesion on her hand. A test is performed, a leptomin test is positive, and a biopsy reveals inflammation of the structure, which has been labeled C in the accompanying photomicrograph. So this is a structure labeled C, and the structure labeled C is which one of the following? Now, first we have to understand the scenario that she has distal anesthesia, and since she has come back from a vacation and her leptomin test is positive, this is a case of leprosy. And in this case, the distal anesthesia, it occurs because of perineural inflammation. And over here, C is labeled, this is the histomicrograph, which has been taken through her nerves, and this is showing us the perineurium. So the correct answer in this case is D, which is perineurium. You can see it is surrounding this whole bundle of axons. And in this scenario, initially there is perineural inflammation, but in later stages of this neuropathy, the endoneurium inflammation also occurs, and demyelination occurs due to infection of the Schwann cells, and that decreases the rate of conduction velocity and causes related further worsening of the symptoms. Let's look at the other labels in this photomicrograph. We know that C is the perineurium. If you look over here closely, D is pointing towards the myelin surrounding an individual axon. E is pointing towards the nucleus of a Schwann cell. And B is the epineurium, which is surrounding the nerve as a whole. It is the dense connective tissue sheath. And A is simply we can see this is the skeletal muscle fiber, which is visible in this photomicrograph. So that was the explanation of this scenario. And this concludes with today's discussion on the histology of the nerves and the ganglia. And I hope this was an interesting discussion of the key points, which helped you understand this topic in a better way. And if you like this, and if you want to watch more of my videos, just like and subscribe to my YouTube channel, AnatomyMentor. You can do that easily by just clicking on my picture, which is going to appear at the end of this video. Also, don't forget to hit the notification button for regular updates on any new video presentations that I'll be uploading on my channel. I also give presentations on different topics from embryology, neuroanatomy, and gross anatomy. So looking forward for your subscription. Thank you. Take care.